Dr. Lee is one of the research recipients of 2017 California Award, I think. He can tell you a little bit more about it. His research is in melanoma. This is Dr. Lee.
Thank you. Yeah. Thank you for coming, and thank you for the invitation. I think the previous speakers give really nice talks. I also learned a lot because I'm not a doctor. I'm just working on some data. Probably my talk is totally different from the other talks, but maybe it's boring for you. But I mean, I try to explain that in the plain language. But I think, even probably you don't understand a lot of these kind of things. But I want to say that from the title that we want to say is personalized drug combination is really important, and also we do need the data analysis to dig out, what's the real effective drug combinations. So as the previous speakers mentioned that we have good immunotherapies coming out right this year, and we have other targeted therapies for BRAF inhibitors. But it turns out, cancer still, especially for melanoma, still a very scary disease, right? We cannot claim we can cure cancer, right? We still need to try our best to find the best drugs and drug combinations to help our patients. So my talk, again, so the title is Personalized Drug Combination Therapy for Melanoma. And basically, the basic idea is try using the big data to help us to find the drug combinations. So my name, again, is Fuhai. I'm in the Department of Biomedical Informatics. And actually, my background is applied math in information science. So this is why I work on this kind of data analysis. So first, so I think you probably know this, right? The previous speaker also mentioned the genetics background of melanoma, right? So this is, I think, one of the most, technology advancement in the, biomedical field. So that means the whole genome sequencing. So 15 years ago, if you want to measure a patient's genome, it costs about $100 million for one patient, for one sample. So basically, there's many countries over the world to work on one project. So basically, we split the data of one patient into different small, blocks, and let many labs over the world to measure what's wrong in the genome. 5 years ago, it's reduced from $100 million to, $10,000. It's a big, significantly reduced, the cost reduced. But today, it becomes amazing. Almost, it takes only, so someone mentioned that it's a rough number of $1,500 or $1,000 per genome. So basically, once we get patients, for example, the tissue, like surgery is always the most important, the first thing we need to do have our patients. Once we get the tumor samples, we send the tumor samples into like a sequence center. In 24 hours, we can get the results of genomics of that tumor. And the cost only about $1,000 or around $1,000. So this is amazing. So it sounds like, we know everything about that patient, about that tumor, right? So now the question is, there's no routine cancer. So every patient's genomic data is totally different. Not totally different, but they are different. You cannot say, okay, this drug works for that patient, maybe it works for another. No, we cannot say that. So this is why we say the precision medicine. So the challenge is, even we have that data, the challenge is we have so many data for one patient. So like mutation data, copy number data, gene expression data, methylation data, microRNA, protein data, and also clinical records data, right? So that means what? Means so we have data, but we don't have knowledge, right? We have data. So for example, every patient have about, we are from a single cell, and in each single cell, there's about 3 billion TCGs, 3 billion. How can you understand the 3 billion things, right? We can't, even the smartest guy in the world probably cannot do that, right? So we have to ask our computer, see whether we can integrate this data together and try to answer to get some knowledge. The knowledge means what? So once we have this patient's data, whether we can recommend what's the best drug for that individual patient. So this is why the TCGA, the Cancer Genome Atlas, screened many different subtypes of cancers, and many patients, they also they acquired all types of data, try to understand what's wrong in the tumor really, and what's the driver things. Okay, for example, the easiest way to understand a genetic cause of the tumor means if we have a mouse, we knock down a gene in that mouse, the mouse will grow tumor automatically. Even it's a healthy mouse, and then once something wrong, genetically it will develops tumors. This is why we believe that genetics is really key for us to first to understand what's wrong in the tumor. The second is, can we find the best drug combination for individual patient? Because there's no routine cancer. I think this is this sentence is from the James Cancer Center. So we talk about the melanoma because I work with Dr. William Carthen to get this peritoneal ideal grant for translational award. So basically, so for example, in general, there are 4 subtypes of melanoma caused by different genetic mutations. For example, BRAF, NRAS, NF1, and Y type. So basically, the last one has no obvious dominant mutation, but this still can develop tumors. So, but this is a gene. So then, if something wrong in this gene, the patient will have a tumor. This is a real driver. Again, if you think about it, there's a healthy mouse, and if you knock down that gene or make something mutation in this gene, the mouse will have tumor. So this is validated. It's the truth. Even that, I think the previous speaker make a slide said that BRAF and NRAS, these 2 genes, actually they are neighbors. They are one's upstream and downstream. You think about it, okay, there are 2 genes and they work together. Suppose, they have, no matter this one or this one, if something wrong in here, they will have a tumor, and if something wrong in NRAS, it also have tumor. But suppose they are same, right? Because they are on the same single pathway. But actually, you can see that these 2 genes cause melanoma, they have totally different, genetic pattern. So that means it's not so easy, not as we understand, say, okay, we understand, okay, these 2 genes work together, in a sequential way. No. There's a lot of things we need to consider rather than these 4 subtypes. In other words, even you have genomics data of our patients, okay, this patient have BRAF mutation, that patient have NRAS mutation. They are totally different. They have different total drug response to even the same type of drug. And even the patients, for example, with the BRAF mutation, all the patients from this subtype, they have also different drug resistance. So that means it's really a difficult thing. So then people talk about precision medicine. Yes, we talk about precision medicine, say delivering the right treatments at the right time. Yes, but we don't know what's the right treatment and what's the right time, right? This is what we need to figure out, and also the doctors need to figure out. So the good thing is, for BRAF mutation, the drug companies develop a drug, Fuhai. So this drug works really well at the beginning for BRAF inhibition, BRAF mutation. A mutation means something wrong in that gene. So for example, it should be a T, a letter T. It becomes C or A, something like that. So this is a published paper say that, so this is the melanoma patient, really scary. And once the patients take this medicine, say it's almost like a normal people, right? And a few weeks later, say it becomes resistant. So that means what? That means even we have some drug can specifically target on a specific target, works very well at the beginning, but a few months or few weeks later, it becomes drug resistant. So that means, so drug resistance is one of the major challenges in cancer therapy right now. So this is the paper published in 2007. This paper actually is wrote by doctors from Massachusetts General Hospital. So usually they believe that so target therapy works really well at the beginning. However, drug resistance eventually develops to these drugs. So what does that mean? So that means we know the genomics, something wrong, exactly know what's wrong in that patient. And we have one drug perfectly target on that target. And then it becomes drug resistant. We cannot, then we don't know what should we do. I talked with Dr. William Carson. He told me that most time we can do nothing. We can only say whether we can try another drug and then try another drug. So by the average survival of melanoma cancer patients is only about a year. If you try another drug, try another drug, try another drug, it's really wasting the time of our patients, right? But we can do nothing. Honestly, we can do nothing right now. So of course, immunotherapy is another good therapy. But we don't know. Sometimes some patients again is really well respond to immunotherapy, but some patients are still not. So that means we have to help our patients who are not respond to immunotherapy to this given targeted therapy. What can we do? So my research tried to say targeting individual resistant alterations or genes doesn't work because even it works for a while, but it will acquire drug resistance, right? We cannot develop another new drug because if you want to develop a new drug, it takes about I think at about $1.5 billion for about 12 years from compound to a drug. So we cannot afford that. No patient can afford $1 billion, right? So and also it's not, you cannot predict it. Even you find a new target, right? You develop a new drug, then what? And then it becomes drug resistant again. The good thing is, I got this number. It's actually $2.5 billion, about 12 years from compound, from lab to a really drug FDA approved. So that means, it's not possible that, we can solve drug resistance for each individual patient. But the good thing is, so I want to say is, this is, I want to say is drug combination, okay? Because what? Because things like this, okay? If you understand this, it's okay. The things like this, if one gene have a mutation, right, have a drug targeting this one, it becomes drug resistant through another path, okay? So for most cases, we don't know which path will reactivate it, okay? But for most cases, we know the neighbor things, always the neighbor things will have something wrong. For example, if this pathway activated, it will cause cancer, right? If you find a drug to inhibit this target, and it becomes drug resistance because of another pathway, but this pathway should have similar pathway genetics around it. So that gives a hint, say whether we can have drug combinations to target all the possible sync pathways to active tumor, right? Because we have so many drugs, feasibility, yes. So first, we know a lot of things about what's wrong in the tumor patients, right? For example, we have 1,000 melanoma patients, right? We can study for each individual patients what's wrong in their genome, right? We can first identify all the possible genetic mutations. And then we put the drugs. For example, if one patient has BRAF mutation, we put BRAF mutation drugs, but also at the same time, we put another drug to prevent drug resistance in advance, right? Then it can to reduce drug resistance. So this is like this. We call it a knowledge network. We integrate all the TOPS data together to form a network. This network represents in a tumor patient what are the possible signaling to activate the tumor. If we have 2 drugs or 3 drugs to perfectly block all these potential signal network to activate the tumor, then probably we can reduce the drug resistance or overcome drug resistance, right? So this is big idea. So we have 2 challenges. First, can we do this? Can we predict all the signaling pathways that can activate the melanoma tumor. The second is which drug we should choose. We know that right now FDA approved how many drugs? Do you know that? Yeah, around 2,000. 2,000 FDA approved drugs. However, you know what? In clinical trial, many drugs, although it failed, but it doesn't mean it doesn't work. Single drug doesn't work. Yes, it's possible, right? We want a drug combination. There are over about 10,000 compounds already passed the safety test. They are safe to use. They failed by FDA because it doesn't work, really cannot help patients get better therapy. So now for us, we can still use these drugs, right, for drug combination study, not just FDA approved drugs. But think about it, 2,000 FDA approved drugs plus even 10,000 clinical trials, that we call it compounds. These compounds is safe, can be used for combination study. But think about it, if there are 10,000, the combination will be millions of possibilities. Okay, mathematically, it's like that. So how can you map millions of combinations to these complicated networks, right? This is what I'm doing, try to. So, because when you talk to a doctor, it always tell you, give me one drug or two drugs, I help you to test. I cannot give him 1,000 drugs to test, right? So, right, the challenge is it becomes like from billions of possibilities, you only can choose one or two, at most 3 to 10, for them to test in their labs, because it's very expensive, right? So now it becomes very computational challenging, but we have to do that. So I will introduce. Oh yes, this is I also put there. So this is, for example, if we have about 2,000 FDA-approved small molecules, at the beginning there are so many compounds will be studied, and about this field, about this number of 10,000, 20,000 small molecules failed, but they passed the safety test. Okay, the drug companies develop many these things. They can be used clinically. Okay, although it's not approved, but this still can be used, especially for cancer patients. They are, who are resistant to immunotherapy, targeted therapy, right? It's allowed to use it. So, using computational method, there are four advantages. First, we don't need a real lab things, right? Wet lab validation. We just need to work with doctors. Say, okay, I select 3 top candidates from millions possibilities, and then can you help whether we can work together to validate it? The second is fast. Computer, right? Is faster and faster. We can really make predictions really in 2 hours, in 3 hours, and also this drug passed the safety test can be used quickly to patients. This is what I want to do. This is basically the background. The first, again, is many cancer patients, even at the beginning, they are respond to drug very well, but it will develop, they will become drug resistant no matter what. So there's no drug work at all. So the second is whether we can find drug combinations to overcome, this drug resistance by blocking multiple targets at the same time. Okay, this is what I want to do. But the challenge is there will be millions of drug combinations that we only can validate a couple, especially for individual patient, right? If there's one patient first, you cannot say we test many drug combinations. We only, we hope we give the patient the best drug combination at the first time that, we don't want to let the patient try again, because melanoma is, again, the survival time is really short, right? We don't want to, waste the time. And this is the basic idea. I want to explain how can we do the drug combination. So basically, this is very simple idea is, if we classify, for example, the 2,000 FDA-approved drugs into different clusters. For example, each drug, this is, for example, some anti-cancer drug, some antidepressant drug, some other types of drugs. Each type of drug have different functions, right? This is the most simple idea. whether we can find 2 drugs for pairwise drug combination from different clusters, then we can significantly reduce the possibility, right? For example, we select one anti-cancer drug and another is antidepressant agents, whether it works or not, right? Then we can because one cluster have tons of drugs, but we only have relatively small number of clusters, right? This is one possibility. This is the most basic idea, okay? And but computationally, it's too simple, and also doesn't work really well. And then we developed a method called the CGNet. And I think this student, Kelly Reagan, we got the Best Student Paper Award from EMEA Translational Bioinformatics last year. And the basic idea is like this. Okay, once we got the patient's genomic data, we try to understand, again, what's the genetic signaling wrong in their patient. So there are always multiple paths. And then we see whether we can find drugs that can reverse the gene expression. For example, this gene upregulated in patients, whether we can find a drug to depress this gene. And then we also combine the drug combination. We combine the signal network with with the drug reverse gene a pattern, and also the target information. So I don't know whether it's easy to translate these kind of things. And then we can prioritize which drug combination is best. Okay, we got a signal network, the real signal network in melanoma patients, and it looks very complicated. And then we want to say for example, these drugs, we studied 633 FDA approved drugs, and we identified these 61 drugs could block their signal network. And then we further divided these 61 drugs into 19 clusters. For example, this is 19 cluster has this is the biggest cluster with more drugs. And then we want to say, how can we, the question becomes, how can we, prioritize these drugs? Then we defined a mathematical equation like this. I didn't put the data resource here, but once we get this data, we can rank this drug combination. For example, this is the top drug combinations. We predicted, this is a prediction score. And these 2 columns are the real validation results. If it's over 50, that means this drug pair can really inhibit the tumor growth, at least compared to normal, at least 50% better, okay? So, for example, from this so many drugs, then we use our method to predict the best drug combinations, and then we found that we got a p-value. That means our method works really well. It's consistent with the experimental result. That means we don't need an experiment. We just use a computer. We can identify the best drug combinations for each individual patient. And then we do the experimental validation, say this is the drug combination. So the bars shows that this is the cell, melanoma cell. If the bar is higher, that means that there's more cells are alive still. If the lower bar means that this drug works really can kill this most tumor cells. So this is the drug combination. You can see that at different doses, this is the doses of the drugs. And it compared to, say, these drugs, it works really well. And also, this is that's how many how many cells alive. This count how many cells died. So basically, you still can see the purple bar is much higher than the others. Means with drug combination, with many more the melanoma cells are killed. In the apoptosis area. This is validation. So this drug combination, especially this drug, is interesting. It's not an anti-cancer drug. And this is the new drug combination we identified in using computation method. There's no one used this drug combination before for melanoma. This is our discovery first. We sent our paper to a very Nature kind of journal, System Biology and Applications, under review right now. So I want to say this kind of discovery is we using prediction and also validation, we can identify, definitely, really, we can identify some new drug combinations for individual melanoma patients. And also, this is another method. I don't think I have time. We want to talk about it. But the basic idea is we develop another method. So for all the FDA-approved drugs, we can identify the top drugs that have been reported by other studies. So that means our method works really well. And this is the validated drug combination for melanoma through the cell line assay. And then, so our prediction are all top 30, all the validated drug combinations are in our top 30 prediction results. So, so I think this is almost the last slide. Is we know so we know a lot of genetics data, but however the reality is what only less than 5% of these cancer patients, benefited by this genomics data. So we try to push, see whether we can use genomics data to really help our patients to overcome drug resistance, especially for melanoma. And yes, that's the last slide. Finally, I would like to thank the 2 students in my lab, and also Kelly, also a student, also collaborator Philip, and also Dr. William Carson. He's the doctor from OSU, and for melanoma. Yeah, and also I would like to thank the Pelotonia grant. I got this grant with Dr. William Carson for 2-year study, $100,000. So I hope whether our method can really help individual patients, because right, you cannot say, okay, we predict the drug combination for all the melanoma patients. We want to see if we can predict drug combinations for individual melanoma patient in a very fast way, no wet lab, only computer. Once you got genomic data, we give you the best drug combinations to, because single drug, again, always have drug resistance, right? Drug combination always can overcome this drug resistance, especially if we combine our drug combination with immunotherapy, we can probably help, our melanoma patients. Thank you very much. Yeah. Yeah.